What if a computer could look at one of your cells, then tell doctors exactly what's broken inside it, and exactly what to change to fix it? Not someday, within the next 4 years. Scientists are racing to build something they call a virtual cell, and the data they need to train it is being generated right now by the billions. Here's what's actually happening, and why it could change how every complex disease, from Alzheimer's to cancer, gets treated. Diseases like Alzheimer's, heart disease, and most cancers aren't caused by one single thing. They're complex diseases. Every patient has a different mix of genetic and environmental risk factors. That's very different from an infection, where there's one clear cause. For decades, medicine has tackled this with guesswork. Pick one gene, spend years testing whether it's the right target, and hope. With tens of thousands of genes to choose from, that approach is painfully slow, which is a big part of why we still don't have real cures for these diseases. 3 technologies have converged in just the last couple of years to finally make this problem solvable. First, measuring. Single-cell sequencing now lets researchers take a snapshot of what's happening inside one cell at a time by reading its RNA, essentially the cell's real-time language. Second, changing. Tools like CRISPR can switch individual genes on or off with precision across the entire genome. Third, AI, the same kind of model that learned to understand human language by reading massive amounts of text, can now learn the language of cells. The twist is that biological language wasn't written by humans. It evolved. We can't intuitively read it the way we read a sentence, but a large enough AI model doesn't need to. The method is simple in concept. Change one gene in one cell, then measure exactly what happened using single-cell sequencing. Repeat that millions of times, and you start building a data set that teaches a model how cells respond to specific changes. One lab has already run roughly 60 million of these experiments, with a goal of reaching 1 billion within 4 years, not in software, but as real physical lab experiments, scaled up using clever barcoding techniques. Separately, other researchers are tackling the same vision from a different angle, mapping where every protein sits inside a cell, since proteins, not genes, actually do the work and are the targets for most drugs. Projects like the Human Protein Atlas have mapped antibody data across nearly the entire human proteome, and newer AI vision models can now turn microscope images into a kind of map showing which proteins cluster together, built directly from data with no human labeling. Combine that data with AI, and you can ask a genuinely new kind of question. Given every diseased cell and every healthy cell from patients with the same disease, what single change, or combination of changes, would shift the diseased ones back to healthy? That's a question medicine has never been able to ask at this scale before. And it's not locked behind a paywall. Some of these models are being released publicly, with global research challenges inviting outside teams to push the field forward together. None of this means a cure is here tomorrow. The first models are still far from accurate, but the foundation, measuring cells precisely, editing them precisely, and now letting AI find the patterns, is finally in place. For diseases that have resisted medicine for generations, that's the first real shot at moving from guesswork to genuine understanding. If you want more breakdowns like this, how AI is actually being used in science, not the hype, just what's real, subscribe to the channel because this story is just getting started.